Alrighty, as you can see, there are 12 petri dishes here on my table, and all of them have something in common, and that is, well, these all contain the Entoloma Abortivum culture, or shrimp of the woods, for a more common name. I first started playing around with these in my one video that I made, what, 2.5 months ago, where I cloned 4 different kinds of wild mushrooms onto agar petri dishes, just to see what they would do, because I like to observe mushroom growth on petri dishes. That's one of my favorite things to do. And the more I got to playing around with these Entoloma Abortivum, the more intrigued I've become, because from all the literature that I've read, it seems that the Entoloma Abortivum, it's generally known, or it's at least kind of agreed upon, that the Entoloma, well, that the shrimp of the woods nodules or weird mushroom growths that we harvest in order to, like, for consumption, are the result of the Entoloma Abortivum having an interaction with the honey mushroom, or Armillaria malea, or something very similar, and that's what puts off those really funky looking growths that we all like so much. So I think this is the one that I showed in my cloning wild mushroom update video, and you can see that right here we have this really cool looking nodule of growth that really does mimic like a small shrimp of the woods. And if you look at all these other dishes here, that's actually fairly common. This is not a one-off. This is, obviously, this is the one I just showed. It has that big nodule right there. This one right here, you can't really see it all that great, but there's a nodule kind of hiding under here, this little fluffy mass of mycelium. There's a big nodule hiding under here. I'm sure there's some smaller ones in here that I can't quite pick out with my unaided eye. There's a nodule here. Even on this little one that just did not fully colonize the petri dish, it still put off a little nodule of like weird growth that mimics that of a shrimp of the woods. Same goes for this one. You can see the nodules right here, right there, right there. This one is just covered in nodules right there and right there. Can't really see all that amazing because of the condensation on the cap, well, the lid, but not a big deal. This one, same deal, nodule, nodule, nodule. This one, I'm sure there's nodules in here underneath this really, really three-dimensional growth. Like, that is pushed all the way up to the lid. That's pretty cool. I like that one. This is one of my favorite petri dishes of this species so far. Going back to the statement about how it's always been kind of agreed upon that the shrimp of the woods that we harvest is the Entoloma Abortivum, either parasiticizing the honey mushroom or the honey mushroom parasiticizing the Entoloma Abortivum. Whenever shrimp of the woods were first described in like mycology textbooks and science textbooks, it was kind of just assumed that the Entoloma Abortivum was parasitizing the honey mushrooms, and that's what was putting off those weird growths. But there's been some debate as to whether or not the reverse is true. You know, like the honey mushroom is parasitizing the Entoloma, and that's what's causing the abnormal cancer-like tumorous growths that we eat from the shrimp of the woods mushrooms. So I figured, well, if that's the case, I want to do a couple little just quick experiments out here. So let me get these off to the side. I don't really need these ones now. These were just here for showmanship, to show all the different petri dishes I have for this species so far. This is still a work in progress. That brings us to these 6 petri dishes right here. I decided, well, here, let's start with this guy, I think, yep, that one, and possibly, yep, that one too. Okay. Let's start with these 4. These are the petri dishes that I inoculated with both the honey mushroom culture and the Entoloma culture. And you can see I have a line that just kind of gives me a gauge. It just kind of gives me a point of reference as to which is which, because, you know, here's the line, middle of the petri dish approximately. There's the Entoloma name, so I know that on this side of the line, this is where I put the Entoloma culture sample, and then obviously on this side of the line is where I put the honey mushroom sample. And I just did that for all 4 of these right here. And I've gotten some really, really interesting results with these that I wanted to share with everybody here. First of all, Entoloma, at least the Entoloma Abortivum, seems to be a really aggressive growing mushroom on, at least on agar. It's really, really prolific, and it takes over the media that it's growing on very, very readily. It's very fast growing too, especially compared to a honey mushroom. Honey mushrooms are really neat looking on agar, but they do seem to be a fairly slow growing species compared to other species that I've dealt with out here, which is, there's no problem with that. That's just one of its properties. A couple of these dishes did give me some really cool results that I was kind of hoping to see whenever I was doing this whole, you know, like inoculate one side with the Entoloma and one side with the honey mushroom thing. You can see that this Petri dish is the one that contains only Entoloma Abortivum samples, shrimp of the woods samples. And this is the one that has that really large nodule of growth right there. And a couple smaller ones as well, but it's mostly just like 1 or 2 like things of growth, you know, like places of abnormal growth compared to the rest of the mycelial colony. Whereas this one did exactly what I was hoping it would do. On this side of the Petri dish, you know, here's the arbitrary line that I kind of drew to show the approximate part of the middle of the Petri dish. This side of that line, I inoculated it with the honey mushroom. This side is the Entoloma mushroom. And you can see right there that the honey mushroom, that little dark area right underneath where my fingernail is, you can see that the honey mushroom did start to grow outwards this way, which is awesome. And then the Entoloma, which is approximately in the very middle of this big colony here, started growing out that way, which is what you'd expect. And they interacted exactly how I was hoping they would interact. You see right here where the 2 colonies met, it is just a line of the growth that you would associate with that of a shrimp of the woods mushroom. It's just a line of really dense, almost like cancerous, aborted mushroom growth. I don't know how else to describe it other than the way I just described it. Whereas with like this guy right here that I was showing, it's just like 1 or 2 nodules on the whole, like kind of distributed randomly throughout the whole colony. Whereas this one is a definite line of the shrimp of the woods nodules that you can see here. Now, I'm not claiming that these are actual true shrimp of the woods. Maybe these just have a really weird growth pattern, or it's hard to tell with the setup that I have out here. But it does seem like, at least to some degree, that the honey mushroom and the Entoloma, whenever they interact, it does seem to at least catalyze the tendency for the Entoloma to put off those nodules of growth that we associate with shrimp of the woods. And this is another one that has the honey mushroom on one side and the Entoloma on the other side. And this one is very cool in the means that you can see that this really beautiful circle of like really like almost perfect circular growth, that's all the Entoloma right there. And then, yeah, under right here, I guess it would be better if I show you on this side. Right here where that dark area is, that's the honey mushroom growth. This was completely taken over by the Entoloma. I'm almost 100% positive. I'm like 95% positive that most of the mycelium that you're seeing, a little bit of condensation, most of the mycelium that you're seeing on this Petri dish is that of the Entoloma. And I'll show you why I think this is mostly Entoloma here in a second with another Petri dish. But you can tell that even with the Entoloma overtaking this entire Petri dish, the presence of the honey mushroom culture, or the honey mushroom, you know, DNA or whatever, present in this dish with that chunk of tissue sample that I inoculated on the other side of the arbitrary line that I grew here, it did seem to make this particular petri dish put off more of that strange nodular growth. But on the flip side, here's another one that I did the same thing, Entoloma on that side, honey mushroom on that side. And while we do have some really, really dense mycelial mass and some really, really dense growth, at least at first glance, there's not really a whole lot of those interesting nodules to be seen. Maybe under all this stuff that's happening here. Yeah, I don't really know what to take home from this guy right here, this particular petri dish, because this is kind of an anomaly compared to these three, because this is another, oh no, where's the other one at? Oh, because this one's kind of the anomaly from these four, because this is also another one, you know, honey mushroom on one, Entoloma on the other side. And this one does have quite a few of the nodular growth patterns that are present in a lot of these Entoloma like samples. There's a nodule right here and right there. You might not be able to see it all that great because these are actually pretty small compared to the other ones. But one thing that I can say about these mixed culture petri dishes, I guess, is that the Entoloma culture really, really seems to like grow more aggressively whenever it's in the presence of a honey mushroom culture. And that's kind of interesting to me. So I thought, what would happen if I took a petri dish that had a decent amount of the Entoloma growth on it, and then another one with a decent amount of the honey mushroom growth on it separately, you know, like there's just a monoculture in each one of those petri dishes. What would happen if I then placed a tissue sample from the other species on top of the actual mushroom that was growing on that petri dish? So in other words, what if I took a petri dish that only had Entoloma abortivum on it, and then I placed a tissue sample of the honey mushroom on top of the Entoloma abortivum colony, you know, mycelial colony, and then vice versa for the other ones. So that's what I did with these two right here. And let's start with this one. This petri dish is one that had a fairly colonized area of Entoloma abortivum tissue sample, and I placed a small chunk of just regular honey mushroom tissue sample on top of that. You can see that right there, a dark spot under here. That's under here. That is the honey mushroom sample that I placed on top of the Entoloma abortivum culture. All around the edge here, these were not present. Like, I wish I would have documented this a little bit better, but I didn't, so you're just going to have to take my word for it, or don't. It's up to you. But once I placed the honey mushroom on top of here, and you can also see that I took a chunk out of here to, I think I used this exact petri dish to inoculate, well, yeah. I used a culture sample from the Entoloma from this one to place on here for this honey mushroom sample, and then I used a tissue sample from the honey mushroom in this one to place on top of the Entoloma colony. And after I placed the honey mushroom sample on top of the Entoloma abortivum colony, all around the perimeter, almost like probably about a little more than half, there's a little nodule underneath this parafilm here. You might be able to like see that, but at least around half of these like nodules of growth grew. And so I got to thinking, does this mean anything? But if you look at this one right here, which is a petri dish that was fairly well established with a honey mushroom culture, and then I place an Entoloma abortivum tissue sample on top of the relatively well established honey mushroom culture, the Entoloma, which is that white part right there on top, the honey mushroom is the black part, or else the really dark brown part, and the Entoloma is the white part. That Entoloma is just completely overtaking this guy. I don't have a high enough sample size to say anything definitive, but from what I'm observing out here, it does seem, at least from what I can see, it is indeed the Entoloma abortivum is what is parasitizing what. So in other words, I do think that the Entoloma is parasitizing the honey mushroom. I don't think the honey mushroom is parasitizing the Entoloma abortivum. Because like I just said, you can see that this Entoloma tissue sample completely outgrew almost this entire, like all this white growth that you're seeing is the Entoloma. So it completely overtook the honey mushroom, and then it put off these really cool areas of growth that we'll look at underneath the microscope here. Whereas this one, which had the Entoloma already on the petri dish, and then I placed a piece of honey mushroom tissue culture on top of that, it did cause the Entoloma culture that was already on the petri dish to put off these growths around the diameter of the circular mushroom colony, but the Entoloma that is underneath that dark piece of honey mushroom tissue is overtaking that honey mushroom. The honey mushroom is not overtaking the Entoloma. If I had to make a guess based on what I've seen out here so far, like I said, this is still a work in progress, and I'm still playing around with these to see what else I can observe from these. It does seem like it is the Entoloma that is parasitizing the honey mushroom and not the honey mushroom parasitizing the Entoloma. And I think that's pretty cool because I never really thought about it much, but just watching these things grow, as aggressive as the Entoloma grows and as quickly as it grows, I do think that it's the Entoloma taking over the honey mushroom, because the honey mushrooms are so much slower growing compared to the Entoloma. So if that's the case, and that probably means that the Entoloma are able to better utilize the honey mushroom as either a potential nutrition source or use it to piggyback off of the work the honey mushroom's already done. Maybe it's kind of taking advantage of the fact that the honey mushroom is already breaking down the like whatever substrate that the honey mushroom is growing on. So the Entoloma will then take over the substrate that it's growing on because it's already been broken down. It's easier for it to digest and grow on. I don't know. This is just a really, really cool result that I didn't really expect to work out the way it did. I'm glad it did because I think it's really awesome. But I don't know. What do you think? Any input? That being said, even though there's definitely something happening whenever these two interact, there's definitely something happening. I think it's just kind of a property, like a common property for these Entoloma abortivum to just put off these aborted fruit forms, like these little nodules of growth. I really do think that that's just kind of a common property that they already have whenever they do interact with honey mushrooms in some form. I do think that that catalyzes the growth of these odd areas of growth. Don't think that the interaction between the honey mushroom and the Entoloma is the sole reason why these things why the Entoloma put off shrimp of the woods. It can't really, at least from what you're seeing here, it can't really be denied that there is something happening whenever these two species meet. So that's pretty neat to me. That's pretty cool. I really like this one right here just because it shows how well the Entoloma just grows basically wherever it wants once it gets established enough. Because this was placed, like I said, this Entoloma tissue sample was placed onto a fairly well-established culture of honey mushrooms, and it just overtook it. It didn't give a crap that it was there. It just did what it wanted to do. Whereas this one right here that had the Entoloma already on the petri dish and had the honey mushroom introduced on top of the culture, the honey mushroom didn't really do a whole lot. It just kind of sat there. I'm sure it did something that you can't really see at the macro level like we're dealing with here. But maybe if you went in here with like a scanning electron microscope or something and actually picked apart to see if you can differentiate different areas of growth between the mycelial strands, maybe you could see something different. But yeah, you can just see from like normal human vision that honey mushroom didn't do much compared to this dish. I do think that the Entoloma is what's parasitizing the honey mushroom. That's what I'm seeing here, at least. I don't think the honey mushroom is parasitizing the Entoloma. And maybe it's not even really parasitizing it. Maybe just kind of using the honey mushroom as a kind of vessel. Well, not really a vessel, but just like, I don't really know how to describe it. Like, the honey mushroom will be growing on a log or something like that, say, or a tree or whatever. That honey mushroom is going to be actively breaking down whatever it's growing on. It's going to be breaking down the lignin and the cellulose in that tree or log or whatever. And maybe the Entoloma is taking advantage of the fact that the honey mushroom has already done a lot of the work by breaking down that substrate. So it can then move in, overtake the honey mushroom culture, or the honey mushroom colony, and then not only grow on the honey mushroom itself, like the honey mushroom tissue itself, but also then take over the growth of the substrate on which it's growing, since the honey mushroom did a lot of the work already. Because, here, I'll show you what I mean. This is why I think that possibly the Entoloma is using the fact that the honey mushroom is able to more readily break down wood and lignin and cellulose, and is kind of taking advantage of that fact, because here are two of my my centrifuge tubes that I showed in my latest video that I filled with sawdust. These are the exact same thing, same exact substrate, and these were inoculated on the same day. One with honey mushroom, as you can see written on here, one with Entoloma. Honey mushroom is actually doing pretty well. It's actually a really, really, really dense growth of mycelium growing down from the top down to the bottom. And you can see on the top here that it looks like honey mushroom tissue that you see on the petri dish there. And here's the Entoloma that I inoculated same day as I did the honey mushroom. And look at this. The Entoloma placed on top of these wood chips, or the sawdust, it died. It could not grow on this sawdust. So is the Entoloma taking advantage of the fact that the honey mushrooms are breaking down or pre-breaking down the wood that it's growing on, and then using that pre-digested, or at least sort of digested wood, or whatever you want to call it, and using that as a grow medium? I don't know. I don't know how else to really put that scenario, but I thought that was really, really interesting that the honey mushroom is growing just fine off of this sawdust, whereas the Entoloma, these, like I said, these were inoculated the same exact day. They were in the same exact incubator. They had the same exact batch of sawdust, like the sawdust that filled these two centrifuge tubes here came from the same batch of sawdust that I like hydrated and mixed up and put in here that I made for these. And yeah, no, the Entoloma species just died. Like there is the there's the tissue sample from the Entoloma, and it didn't do anything to this sawdust. It just sat on top, and it just kind of crapped out and died. So, yeah. Whereas, like I said, with this guy, honey mushroom is doing fantastically. It loves this sawdust. I don't think that the Entoloma lack the ability to break down cellulose and stuff, but I don't think that, from what I'm seeing here at least, Entoloma don't really like to grow on wood that much. They like to grow on like leaf litter and stuff. But a lot of times whenever you find Entoloma, or at least shrimp of the woods, they are growing usually at the base of a tree. So that suggests that they're growing off the wood. But from my little experiment here, and this is only a sample size of one so far. Like I said, this is a work in progress. It just seems like these Entoloma don't really know what to do whenever they encounter fresh wood without having anything done to them. So, yeah, I don't know. I don't know how else to word that. Just to say the way I've been saying it, like maybe they're kind of taking advantage of the fact that the honey mushroom has already pre-digested or started to digest whatever tree or woody substrate that they're growing off of, and then it's taking over not only the colony of the honey mushroom but also the substrate on which it's growing, and it's kind of using that to do what it needs to do in order to, you know, repopulate itself and proliferate and reproduce and yada yada yada. Very cool. Very cool. Yeah, I didn't think that I would really get into these two species the way I have, but I really like kind of playing around with these two because these are actually really, really interesting to me. Yeah, let's take a look at this guy underneath the microscope and see what it looks like, because I'm really interested in the middle of this petri dish because it has some really interesting looking little bulbs and weird areas of growth. I just want to see what it looks like underneath the microscope. If I have to take the top off of this one, I will. It's not really that big of a deal because I can always do this stuff again if it gets contaminated. But yeah, let's go take that over and look at it. Alrighty, I just looked at this underneath my microscope real quick to see if I had to take the top off. Thankfully, I don't. We are going to get a clear enough picture with the lid on, which is nice because then I don't have to expose this to the air and potentially contaminate this. That's fantastic. There's a few areas of growth I want to focus on. One area is right here underneath my fingernail. I want to check out this area of growth right here with all these little protrusions coming out. They almost look like little mushroom pins right there. And then this guy right there. And I should probably note that all of these are parts of the honey mushroom culture that we're going to be looking at. At least that's what they started out as. If anything that you look at on the screen here is darker, like black or almost black, that's a pretty good bet that we're looking at a chunk of honey mushroom culture. And if it's white, then we're going to be looking at an Entoloma culture. So let's start with this guy right here. All right, that's really cool. It's really neat looking, actually. So yeah, you can see that right here, we're just basically going to say that this is one big circle, at least at this scale magnified. This big circle of growth here is the honey mushroom tissue, and yeah, it looks like that is not Entoloma. It looks like, you see how this is like really like fluffy, like these parts right here are really fluffy. They're like going poof, 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 poof up. And then you can also see throughout this whole section here how we have these really long strands of mycelium here. The really long strands that you're seeing are almost certainly the Entoloma. And these little wispy parts of mushroom growth are probably the... Here, let me get something to point at with... Sure. Because I think my finger is messing with the autofocus on my camera. But yeah, you can see that these really long strands of mycelium right here, and all over really, that's almost certainly the Entoloma. Because like I said, the Entoloma is a really aggressive growing species from what I've observed. And whereas on the opposite end of the spectrum, the honey mushroom, even though, which is really kind of weird because honey mushrooms are a really, really notorious like pathogen, like a pathogenic fungus for a lot of tree species. But they do seem to be a pretty slow growing species at the same time. So it's kind of counterintuitive, but I don't know what to really make of that. That's not really the purpose of this video. But yeah, let's go back to these areas of growth right here that look almost like cotton balls or something. They do seem to be originating from this very dense cluster, or this very dense area of honey fungus growth. And I do think that this is honey mushroom culture trying to either grow out of the influence of the Entoloma, or maybe it's just trying to put off some kind of fruit body. I'm not sure. But that's actually a really, really cool area of growth right there. Let's go to... Now let's turn this and focus on the area that's basically right in the middle of the petri dish that I pointed out. Alrighty. Wow, that's cool. That's really cool. Okay, once again, darker part that you're seeing here on screen. Honey mushroom. All these long strands of mycelium. Almost certainly Entoloma. These two pieces right here look suspiciously like mushroom pins. I can't say with any certainty at this point whether this is an actual set of mushroom pins, but from what I've seen underneath my microscope with other species of mushrooms, these do look like mushroom pins. So I think, I'm just kind of guessing, I should say, I think that these are honey mushroom pins that this culture is trying to put off fruit bodies. Even like not magnified. Here, let me take this out. Let me turn the flash on so you can see. Come on, focus down. See those? They look like really tiny beginning stages of mushroom growth. And they do not look like Entoloma. I'm gonna turn my flash back off. That is what you would expect to see from a primordial mushroom, like kind of like the in-between stages of Primordia, which is the stage that a mushroom colony goes through before it puts off the actual mushroom pins. It's like the step between mycelium and mushroom pin, if that makes any sense. So I'm almost positive these are honey mushroom pins. That's cool. So let's go to that last section that I talked about and look at that, because I think it's more of the same. Yeah, once again, dark area kind of ends right about here. So the honey mushroom culture, at least this dark part of the honey mushroom culture, ends right about here. So you can see all this dark pieces under there. That's definitely part of the honey mushroom culture. And once again, this almost looks like Primordia of a mushroom colony, of a mushroom culture that's trying to at least initiate fruiting. So I think that this honey mushroom culture is trying to put off some fruits, or at least do its best attempt to put off some fruits given the circumstances. And I know that may seem kind of improbable because it is being completely overtaken by the Entoloma, but I have observed that a lot of mushrooms, whenever, like say you have something like a culture growing on like grain or even a petri dish, if that culture or if that colony of mushroom tissue or that starts to be overtaken, or if it encounters a contaminant, that culture of mushrooms will then attempt to put off some type of fruit as a last-ditch effort to proliferate and try to put off some spores to at least attempt to carry on its genes. Like, even if a mushroom culture in a ball mason jar or whatever gets contaminated, sometimes it will still put off fruits as a last-ditch effort, kind of like a now or never thing. Like, if we're going out with a bang. It will still put off fruits. And it almost looks like that's what's happening here, because none of the other honey mushroom samples that I have out here on petri dishes are doing this. It's only this one with the Entoloma tissue, with the Entoloma species completely overtaking it, where it's trying to put off what looks like, I guess I should say, what looks like primordial growth. Oh, that's neat. Huh. Oh, I was going to say, oh, that's really cool. Look at that. Look at those rhizomores. No, that's just the writing that I have on the top of the lid. Yeah, Entolo. Okay, never mind. But yeah, no, like, I really do think that both of these species in this single petri dish they're definitely affecting one another. I mean, that's a given, right? It's, you put two different things in a petri dish, they're going to compete for resources and they're going to interact with each other in some way. But yeah, it looks like the Entoloma is making the honey mushrooms prematurely put off fruit bodies, and whenever the Entoloma encounters whenever the Entoloma encounters a honey mushroom culture, it makes it kind of catalyze that reaction. Well, it kind of catalyzes the event of it putting off those weird aborted nodules of growth. While we're at it, let's get this guy that has the honey mushroom tissue on top of the Entoloma colony. Let's see what that looks like under here. I think this is about as good as we're going to get here. But yeah, this obviously darker part is the honey mushroom tissue that was placed on top of the Entoloma colony. And you can see that it's been completely overtaken by these Entoloma strands of mycelium. The honey mushroom doesn't have a chance. The honey mushroom seems to not stand a chance against these Entolomas, at least in the lab setting here. And I obviously use the word lab settings, well, the words lab settings very loosely in this context. But yeah, there seems to be no real indication that the honey mushroom is overtaking the Entoloma or growing onto the Entoloma. It looks like the Entoloma is completely overtaking the honey mushroom. So from what I'm seeing, it does look like what was originally thought, like whenever the Entoloma abortivum was first described, was, you know, the thought of it's the Entoloma is parasitizing the honey mushroom. I do think that is about what's happening here. It's probably a lot more complex than that, but I don't think that the honey mushroom is the one that's doing the parasitizing. I think that the Entoloma is the parasite in this context, and I think that the honey mushroom is the unfortunate host, I guess you could say. It's the losing party in this interaction is the best way to put it. Very cool. And yeah, let's look at one of these nodules here too, while we have this underneath the microscope. Yeah. Yummy. Yeah, and see, that's definitely Entoloma growth right there. And just like I've seen with a lot of these other petri dishes, especially magnified like this, they just look like mini shrimp of the woods. They're a little darker than the shrimp of the woods that you get out in the woods, just because these have been growing for so long in like a controlled environment. They seem to not have to. They don't have to worry about like getting taken over by bugs or other molds or insects or like bacteria or anything like that. So they are just doing what they want to do and be happy. Huh. Very cool. So yeah. Let's go back over here. So I guess the takeaway that I'm getting so far, like if there's any updates that I like observe in the future, I will give you that update. But from what I'm seeing out here, it looks like what was originally thought to be the case is the case. I do think that the Entoloma is what is either parasitizing or taking advantage of the situation of coming in contact to the honey, like with the honey fungus, and using its resources that it's already freed up through its digestive processes. And I think that is what's catalyzing the very large growths that we get from the shrimp of the woods mushrooms out in the wild. But on the flip side, I do think that it is commonplace for these Entoloma to put off aborted fruit bodies all on their own. It probably doesn't happen like this out in the wild too often just because there's so many variables out in the wild. But I don't think that the interaction with the honey mushroom is the only cause of these aborted fruit forms, like the shrimp of the woods. This is really interesting. I got to do more with this stuff because this is so cool. But for now, this is just the findings and the observations that I've made. None of this is, like, I'm not claiming to have discovered anything like remarkable or anything like groundbreaking in this field or between these two species. These are just based on my observations. But this is really cool. All right, I just wanted to show you this stuff, so this video is probably long enough as is, so I'm going to let you guys go, and I appreciate you watching. I'll see you next time. Bye-bye.